So I want to thank the organizers for inviting me to talk today about some of our recent efforts on developing a new platform. I'll start off with a cliche slide that you've all seen before, and I really only want one brief point off of this, is that we're really trying to push down. There's still a lot of work to be done to push to the bottom of the pyramid. And the point, obviously, to get there is to see more patient impact, see more people sooner, and be able to get them on appropriate therapy. And when we're talking about microscopy centers, this is one we visited in Uganda. You can see this is probably one of the nicest ones around because of the nice, clean biological safety cabinet up in the upper left. They actually are very variable, and probably many of them in extreme conditions, harsh environments. There was just a paper Madhu had mentioned this morning that he and Claudia Denkinger have published looking at a survey across the world. And in most cases, it's a lack of power, it's a lack of water, it's a lack of skilled workers, space, etc. So really it becomes a very challenging environment. But this is where we needed to develop an assay that worked both for case identification and for drug susceptibility testing, because this is really where the need is, and we wanted to try and drive our development to that location. So some of the key benefits of the test that we've developed are the only infrastructure is intermittent power, so it can run off a complete day off of a single battery charge. It does need to be recharged at some point, whether that be from solar panels or a car charger or an unstable electrical grid in most cases. It's fully integrated, it's self-contained, so it has very high precision and consistency, and it's very safe for test operators because there's no safety equipment necessary. We took as a starting design principle, to use the entire full volume in order to get the best possible sensitivity on the possibacillary samples. And so that's something that we really just decided that we wanted to do, is try and drive that performance as best as possible. We've optimized the workflow with just using best-in-class technologies. Nothing that's extraneous is included. So we were charged with pursuing an aggressive development timeline of 24 to 30 months from our commitments with the Gates Foundation. And in order to accomplish this, our strategy for doing this is to take some existing technologies that we had just developed, cutting-edge nascent technologies. This is, on the left, is the isothermal amplification technology developed at Ionian, and try and integrate that chemistry onto the new Alere Q instrument platform shown on the right. And this is a fully capable quantitative real-time PCR instrument. It's fully portable. And what we wanted to do is, you'll notice the cartridge is very small, and currently the first test coming out on that is for HIV 1-2 viral load, and that uses in the first instance a small amount of whole fingerstick blood. So the difference in trying to detect active pulmonary TB is that the sputum volumes are much larger than that. And you can see there's actually quite a bit of discrepancy all over the world. This is just a simple Google search of different sputum containers. There's quite a bit of variability out there. There's not really any standardization. People use all kinds of different containers, and some of them are really quite unsafe for the patients as well as for the test operators. And so we wanted to try and change this. And what we've come up with is, how do you then attach a sputum collection cup to the cartridge? And what you'll see here is the cartridge that we've come up with. On the left-hand side is a full sputum cup, and it actually seals properly. And not only that, there's the cartridge on the right that matches the same shape and size of the existing cartridges, so that we've now married effectively sputum collection with sample processing and detection all in the same one piece. And so there's no handling done by the test operator whatsoever, no transfer steps at all. And then, of course, the other challenge is that now that we've stuck this large sample cup on the outside of the cartridge, it doesn't fit into the original instrument. So we've had to redesign the instrument in order to be able to accommodate these larger volumes. The important point to note here is that this new instrument is fully backwards compatible, continues to run all the existing cartridges. It just now has the ability to take not only small volume tests, but also large volume tests that can be sputum or it could be other matrices. So just to briefly cover exactly what's going on inside the workflow inside the cartridge. This is looking down on the top of the cartridge, but of course as the sputum cup is attached to the cartridge, you're looking up through the bottom of the sputum collection cup on the left. And you can see these notable paddles here. These are used when the instrument injects the liquefaction reagent and provides a fully automated liquefaction homogenization, provides a very consistent, reliable result. It can take any volume of sputum from just hundreds of microliters up to 10 mils, which would be an exceptionally large volume. So it can handle all of that, and it pumps the entire volume into the cartridge. And down in the depths of the cartridge underneath this label here, there's a full nucleic acid purification and enrichment for mycobacterial DNA. And after that, it then pumps over to the reagent reformulation where it hydrates. And of course, these cartridges have to be stable at high heat in extreme environments, so they can handle 40 degrees C easily. And the reagents then rehydrate in a matter of seconds. It gets pumped onto the reaction chamber, where in the case detection, it's isothermal, and in the DST, it's PCR. And then there's amplification and real-time detection. So first off, is important is to identify those individuals who are in fact positive for TB, and only maybe 10% of your TB suspects will turn out to be positive. So we wanted to come up with a strategy of how we think drug resistance testing should be done, and obviously it's open for discussion and welcome input, but we'd like to try and match the clinical utility to the drug resistance information to the clinical utility and the location that's needed. In other words, when a patient comes into a microscopy center or somewhere else, they first get identified as, yes, they have confirmed TB, and secondly, they get tested for first-line drug resistance testing on those individuals, and then for those individuals who are susceptible to first-line therapy, they can be administered therapy right there at that time in the location. And for those who are either MDR or other resistance, they would get referred typically to a center or a location that could handle those types of patients, and then there would be available second-line drug resistance. There's of course no limitation on where these testings can occur. They can all occur at any place that the instrument works, because it's all the same cartridge with the full automated sample prep. It works off of PCR amplification and a competitor-monitored amplification detection scheme. That's an Alere chemistry for detecting on a array, which is ideal for multiplex SNPs. And I'd like to stress that right now we're developing a model system so that we can then develop the cartridges, the instrument, the platform, and then modify the arrangement of SNPs based on the outcome of the upcoming trials on different regimens, different therapies, so that we can match what is on which cartridge to the actual clinical practice. So just, this is a quick summary. You're probably already familiar with, from the literature, looking at the different drugs and gene loci and the important SNPs. So this is rpoB, CATG, INHA, ethambutol, and then on again to GIRA for the fluoroquinolones and the injectables. And we may switch these around depending upon the outcome of those results. But we want to develop a platform first, which works like this. So it's the same cartridge, of course, but instead we have a PCR amplification and then microarray, and the instrument takes an image of this, and these different spots then change fluorescence over time, and this can be extracted in the lower right. You'll see the decrease in fluorescence of the different spots, and based on that we can then infer mutant versus wild type. And this whole process takes only 30 to 45 minutes depending upon the specific assay. We're looking, basically here is an example from rpoB, looking at plotting the initial versus the final signal, fluorescence signal for both wild type and mutant, and you can see in each of these different examples by plotting them, you get a nice tight cluster of wild type and mutant alleles. And so in every case we have an easy discrimination of which are the mutants. So to summarize, we've been challenged with a technology product profile for the development of a high-performance NAAT. Of course, I want to emphasize that this test is designed for use in really distributed low-resource settings, and particularly microscopy centers. It's a technology that enables point-of-care testing, provided that it is properly put in place in a program. We have aggressive development timelines, and so in order to accomplish that, we've been merging two existing technologies. This provides fully automated sample-to-answer processing for TB diagnosis, first-line and second-line drug resistance testing. I need to quickly acknowledge many people are working on this, but certainly my affiliation with the program officers at the Gates Foundation, Boris, Jean, and Jim. And thank you.